So we're a part with this 9 and AViDD program supported by NIH, led by David Perlin and Charlie Rice. Our project is focused again on the inhibiting of the RNA synthesis of the variety of RNA viruses. First, I'd like to thank all the researchers from our laboratory at Columbia University for contributing the best efforts into this project, and our collaborators, Dinshil Patel and Tiago Moreno, Tom Tuchel, and Robert Kursdorfer. So during the pandemic, we initiated the project, and then all the cores and the AViDD Center and the general support by the NIH. So our research is focused on studying the interaction of the nucleotide analogs and the polymerase. We're designed synthesis novel nucleotide analog and study their interactions with the polymerase. So then we can sort of use this kind of expertise to pursue two different projects. One is develop, leading sequencing technologies. The other is to develop antiviral drugs. I use the sequencing technology to first demonstrate the power of this approach. So for example, we use structure design, a directed approach, design these nucleoside triphosphate reversible terminators that led to sequence the human genome from the Sanger era sequencing to a $1,000 genome. Essentially, these molecules allows the entire genome to be deciphered on the surface of the chip. So this is a technology that is like over 70% worldwide sequencing uses this approach. So again, this general approach of rational design, these nucleotide analogs, applicable for sequencing technology development based on polymerase reaction. Also, I'm going to show that antiviral inhibitor development. So for example, for sequencing, again, we target polymerase. Here's the template, here's the primer, here's the incoming nucleoside triphosphate, and complex by the polymerase active site. So again, our focus on these molecules. For sequencing, we need to make sure the molecule is recognized by the polymerase. Over here, we realize there's not much room, so we need to use a small and inert reversible blocking group. And then over here, there's a larger gap, therefore, so we can design a cleavable linker fluorescent dye. It turns out this molecular insight is critical for developing these molecules. Then this was first generation of this cleavable fluorescent nucleotide. You can see it's very complex. However, so we have the expertise to design synthesis of them, and then these allowed the sequencing reaction be performed in the polymerase reaction on the surface of the chip. For example, the perfect incorporated nucleotide incorporated reaction stop by the color. You can detect the sequence. Then it removes fluorescent dye and generates OH group and continue this process on the surface of the chip. You can decipher the entire human genome on the surface of the chip with $1,000 or less. But over here, right, it stopped the polymerase reaction. Over here, we need to continue the polymerase reaction. So this, technically is much more challenging than you stop the virus polymerase reaction, because the virus polymerase reaction stop. We just need to pursue this step. So that we use once the pandemic started. So we thought use our expertise first to design and screen these RNA synthesis inhibitors for coronavirus. So my talk is going to cover two aspects. One is to deal with inhibit the coronavirus RNA synthesis. The other is to inhibit the RNA synthesis of dengue and chikungunya. So first, I'll focus on this coronavirus. SARS-CoV-2 has a RNA-dependent RNA polymerase and also has an exonuclease, which both are critical for viral genome replication and transcription. So the exonuclease removes certain nucleotide inhibitors incorporated into the viral RNA, so therefore decreasing the efficacy of this drug for treating COVID-19. So this poses a challenge for nucleotide analogs to inhibit the coronavirus. So we are pursuing two approaches to inhibit RNA synthesis of coronavirus. First is combination of two inhibitors. One inhibits RDRP, the other inhibits exonuclease. The other is to design and synthesize novel nucleotide analogs that once incorporated by the RDRP, it inhibits the further RNA synthesis and also resist exonuclease. Here, we don't need to directly inhibit exonuclease, basically bypass the excision because the exonuclease cannot remove the nucleotide because of its unique features. So both of these approaches will efficiently reduce replication of coronavirus and related virus. So again, I focus on the RNA synthesis part of coronavirus on two enzymes. Again, one is this RDRP polymerase. The other is exonuclease, right? So both of these are sort of working in tandem to synthesize the larger RNA genome of the coronavirus. To inhibit the RNA synthesis, we really need to target both. So the first approach is a combination of two inhibitors. One inhibits the polymerase and one inhibits the exonuclease. So here we have demonstrated sort of the proof of principle at molecular level and also in vitro at viral level to have inhibited. So first I'll show present the assays, molecular level assays we developed. One to evaluate inhibitors in polymerase reaction and the other on exonuclease. So at the beginning of the pandemic, so we established collaboration with Robert Kirchdorfer at University of Wisconsin Medicine. And then we developed this simple and powerful molecular assay, where you have simple template, the RNA primer. And then we add this nucleoside triphosphate. For example, we identify the triphosphate of sofosbuvir, actually a terminator of SARS-CoV-2 polymerase. And then we use multi-top mass spectrometer to measure the reaction efficiency, so on and so forth, because as the mass spectrometer give our molecular level details. For example, this is starting primer. Once it's nucleotide incorporated terminated, you see a single peak, and then you can measure efficiency, so on and so forth. So it turns out for coronavirus, this triphosphate, sofosbuvir even is able to terminate the RDRP polymerase reaction. However, its incorporation efficiency not able to compete with the natural UTP, and also because exonuclease can remove the incorporated nucleotide. Therefore, this type of sofosbuvir has a little inhibitory activity for the coronavirus. So we also, again, identified sort of initial molecules which are the hepatitis C NS5A inhibitors. These all FDA-approved drug hepatitis C. And it turned out that these molecules also inhibit SARS-CoV-2 with different potency. So these sort of the exonuclease assay we use to evaluate the inhibitory activities, how to deal with the exonuclease innovation. So here, we start with simple RNA substrate, for example, and then you add exonuclease. You can see these paired molecule RNA are degraded very rapidly, turn into small fragmented peaks because exonuclease remove one nucleotide, after another, so from the 3'. But now, if you add these inhibitors with increased concentration, you can see, right, that most of the RNA are protected because it's this molecule inhibits the exonuclease activity. It turned out that danclostevir also inhibits the RDRP activities. For example, here, again, we use a simple polymerase reaction at polymerase and a simple substrate. And here is the starting RNA substrate, here extension RNA product. Again, this is measured by mass spectrometer. So when you increase the concentration of danclostevir, you can see, right, the extension product is reduced. So we still need to identify the mechanism of this molecule because it's not nucleotide based. So Again, here is an overview of the exonuclease assay. So basically, so we can use this kind of screen to identify nucleotide analog that once incorporated RNA, whether it resists exonuclease or not, and also we can use this assay to evaluate the potency of the exonuclease inhibitor. Again, here is a parent molecule, a single peak without exonuclease. Once we added the exonuclease without any inhibitor, you can see this natural RNA are rapidly degraded. So once we increase the concentration exonuclease inhibitor, you can see this parent RNA is protected. So, remdesivir is sort of the first approved COVID-19 drug. However, it has has not much very high potency in the clinical setting. So the mechanism of sofosbuvir inhibits coronavirus are described as follows. One is called delayed termination. Basically, once remdesivir incorporated, and then there's another natural nucleotide incorporated. Presumably, this will invade the exonuclease. The other is that the remdesivir actually get into the viral genome, while the virus replicate, and once the viral genome RNA contain the remdesivir, and then the replication, the synthesis of complementary strand is inhibited. So if both of this sort of a mechanism works perfect, right, then the sofosbuvir should be a very potent drug for COVID-19. So, however, we showed later on that the sofosbuvir terminated RNA has no exonuclease resistance. But one beneficial aspect of sofosbuvir, right, when it turned into the active triphosphate, is this incorporation, high efficient incorporation by the SARS-CoV-2 RDRP because of this cyano modification. So this is structure determined by Elizabeth Campbell's group at Rockefeller. So the identified because of this cyano group, there's additional hydrogen bonding formed. Therefore, this bond very tight to the active side of SARS-CoV-2 RDRP, even better than the natural nucleoside triphosphate ATP, because sofosbuvir is remdesivir. Remdesivir is an analog of ATP. So then we synthesize the RNA. They may make the delete termination. For example, here is remdesivir, and here is the 3 natural nucleotide incorporated. So this sort of a delayed termination product. And here is the natural RNA. And then sort of we treat it with exonuclease. You can see once you add exonuclease, so all the parent molecules, the natural one, rapidly degraded. The same with this remdesivir terminated RNA, rapidly degraded. However, we add exonuclease inhibitor. You can see all these RNA substrate are protected. So if this also works, in the virus, then we combine remdesivir with exonuclease inhibitor. It should improve the virus inhibitory activity. Again, our collaborator Tiago Moroso in Brazil performed this viral synergistic assay. For example, remdesivir itself has this much potency. When we combine with this exonuclease inhibitor, you can see the potency increased tenfold, and so on and so forth. So now I'm going to switch gear to move to the sort of the second strategy, which is to design a synthesized nucleotide analog with enhanced SARS-CoV-2 RNA RDRP reaction termination activity, but also resist exonuclease, which is the nucleotide itself terminate its RDRP reaction, also resist the removal by the exonuclease. So the molecule we started with is this tenofovir. So we synthesized a RNA terminated with tenofovir. You can see tenofovir does not have any hydroxyl group is a select sugar, and here's the natural RNA. It turns out when we add exonuclease and natural RNA are completely degraded, but however, this type of RNA terminated by tenofovir is highly resistant to the exonuclease. But here is the challenge, but this type of molecule had very low incorporation efficiency by the SARS-CoV-2 RDRP. So we designed a synthesized variety of the RNA terminated by these different nucleotide analogs with different features and identified a trend. It turned out this type of nucleotide analog have high resistance to exonuclease resistance. So then from it also constructed a model of the RDRP complex with this nucleoside triphosphate substrate to explain why this tenofovir has reduced RDRP incorporation by SARS-CoV-2 polymerase. You can see here the natural substrate ATP from a very tight, complex and is active side of the RDRP and from this hydrogen bond. And then here, the tenofovir has methyl group. It's hydrophobic, so it's incompatible with this polar amino acid residue. Therefore, this has a very low activity for incorporation by the RDRP. But based on this struggle, we thought, okay, how about remove this methyl group, right? So this and turn this adofovir. You can see that you release the clash, and then it should be incorporated better by RDRP. And our experimental data showed that this actually indeed the case. So another way to look at this is that we can evaluate another isomer. So with this methyl group turn the other way, this also relieve this kind of clash. Further on, if you put a polar modification with this fluorine, this compatible with this polar amino acid, the active site of the polymerase, and turns out this molecule has several hundred-fold increase in terms of RDRP termination efficiency than tenofovir. So again, this is the result. So this is the natural nucleotide. You can see this fluorine modified tenofovir has pretty high incorporation efficiency by the RDRP. And then this kind of a molecule, when incorporated RNA, it also resists exonuclease. So it's a natural RNA, the exonuclease completely degraded them pretty fast, and this molecule has high resistance. So now this is a summary with this kind of structure-directed rational design approach. We're able to increase the RDRP termination efficiency over 300-fold, whilst this molecule incorporated RNA still maintain the resistance of exonuclease. So we're designing and synthesize the prodrug this molecule and move on to the in vitro virus testing. So now I'm gonna get into the aspect of the exonuclease cleavage and resistance. For example, over here, this is sort of the active side of the exonuclease. Exonuclease SARS-CoV-2 remove nucleotide from the 3'. So here is a phosphate bond, and then this can be broken by the exonuclease very fast. But however, if we replace this oxygen with a methylene group, right, you form a phosphonate, and this is a much stronger bond. So this should slow down the reaction. This kind of modification we expect will sort of resist exonuclease. So all the tenofovir analogs have this kind of features. So now we like to combine this kind of phosphonate modifications with the remdesivir modification on the cyano group, right? Cyano group modification increase the RDRP incorporation efficiency. Actually, this remdesivir incorporate better than the natural substrate. So again, the phosphonate modification theoretically can enhance the phosphorylation efficiency of the prodrug to the active triphosphate. For example, the remdesivir, the prodrug, get into the cell. It first need to turn to monophosphate. However, this reaction is reversible. This phosphate can be lost, turn back to nucleoside. So therefore, it's not very efficient to turn into triphosphate. So if we replace oxygen with the methylene group, and then this phosphonate group is not reversible, so it can be more efficient to turn to the triphosphate substrate to inhibit the polymerase. Again, these some examples of the molecule we designed synthesized. This is a prodrug. Again, here you replace with this one with methylene group. We call this sort of a methylene RD still based on this remdesivir scaffold. It turns out, so this has reduced activity for coronavirus. But however, when we evaluated this for chikungunya virus, so the remdesivir has absolutely no activity for chikungunya virus. But however, we just replace the oxygen with this methylene group. You can see it has very potent activity for chikungunya virus, target the RDRP of the chick. And then if we replace this oxygen, 3' OH with amino group, and then we turn remdesivir into an obligate terminator, and this molecule has potent activity to inhibit dengue virus. For example, this is the potency of remdesivir itself, and these are the analog, modified analog, more than 10-fold potency. So we also evaluated these prodrugs in 2 different MOI. In both cases, the potency increased compared to remdesivir over 10-fold. Subsequently, our collaborator Dinshaw Patel's group cloned the RDRP for dengue. We showed that this molecule is an obligate terminator of this dengue RDRP. So we also evaluated another prodrug. We synthesized based on this 3'-deoxy-A. This 3'-deoxy-A is an obligate terminator of the RDRP. It turns out this kind of molecule also has potent activity to inhibit chikungunya virus in vitro in both MOIs. So now we'll move on to synthesis of the additional analogs and also change the A base to the others to evaluate their inhibitory activity for the RDRP and synthesize the prodrugs to inhibit the virus. So we also sort of evaluate a variety of these prodrugs with the aim to improve the in vitro and in vivo pharmacology parameters and potentially develop these early lead drugs into oral therapeutics. Thank you very much for your attention.